Friends, this Friday in Time with Thebsian, I will be talking to you on USP Testing of Plastics. That is, testing methods for plastics as described in the United States Pharmacopoeia and National Formulary of United States. This is USP is a Bible for the manufacture and testing of drugs and food in United States. They have a chapter, if I remember it is 88 or something like that, just for testing of plastics used as containers for food, drugs and solutions used in drug. USP testing has become very strict and trustworthy battery of tests. Here, plastics are tested for their compatibility for non-toxicity and safety. Because we can see that many of the eye drops, cough syrups, many other drugs and food are stored or packed in plastic containers. So many different type of plastics are used for making containers. There are cheap plastics, there are very costly plastics which are very safe. But to make sure whatever plastic is used by a manufacturer, they have to undergo testing using USP standards. So the general conditions and tests that are described in United States of Pharmacopoeia for plastic. In USP tests, there are 6 classes, and these are used to certify that there are no harmful reactions or long-term bodily effects from various plastics or materials used as containers. So as I mentioned, these tests are used for plastics coming into contact with human body also, because plastic blood bags, plastic saline bags, IV sets, these are all made up of plastics. So actually they have to undergo the USP testing. So the saline for injection, especially saline for diluting various drug for injections, common plastic bottles only. In USP has a test solution and also control solution. Test solution will have the extract of the test material, but same extract without test material is the control solution, or it is treated as the negative control. Likewise, laboratory animals are used for some tests. Whatever may be the control samples or controls are used as per USP standards. Class 1, let us see what tests are being given to be carried out so that the materials can pass the class 1 test. Test materials are extracted in 0.9% sodium chloride injection, and they are injected at a dose of 50 ml per kg intravenously into a laboratory mouse, and the same extract is injected intracutaneously on the dorsal side of the rabbit, 0.2 ml per site. They are injected at 5 sites. And at another 5 sites, control injections are given. Control injections are, as I said earlier, only the only the extracting medium. The extract of the test sample is the test solution, and simply the medium alone is treated as control. So, in the case of acute systemic toxicity, 5 mice receive control injection. Class 2, other than the class 1 extraction tests, the tests are carried out 1 in 20 solutions of ethyl alcohol and sodium chloride injection, and they are injected as for the saline extract intravenously in mice and intracutaneously on a rabbit. For class 3, in addition the tests required for class 1 and class 2, another extract also is made using polyethylene glycol 400, and the mice are injected at a rate of 50 ml per kg intraperitoneally, not intravenously. Rabbits, 0.2 ml is injected intracutaneously. For class 4, another extract using vegetable oil, refined cottonseed oil or sesame oil, IP grade is used for the extract, and they are injected like the polyethylene glycol intraperitoneally in mice and intracutaneously in rabbit. Paravertebral muscle of the rabbit is prepared, and 1 into 1 into 10 mm size strips are implanted in rabbit paravertebral muscle, or same strips are implanted in subcutaneously in rats. Class 5, devices have to undergo all USP tests designated for class 1 to class 4 except implantation test. Class 6, device has to undergo the tests designated for USP class tests. This is most stringent, and most of the modern device makers make sure that their devices, their materials for preparation and the end product undergo the USP Class 6 test. The reason that they want to assure that their products are safe. If you look closely, you can understand that in the USP tests are of 2 types. One is extraction, another is implantation. For extraction tests, there are specifications for specimen sizes. Samples can be powder strips, sheets, and granules. Samples are selected as per the direction given in the USP. In general, criteria is based on the area of the sample used for extraction, and that is based on the thickness of the sample and the area of the test sample. For implantation test, the ideal sample size is 1 into 1 into 10 mm size. When it comes to the procedure, the toxicologist has the benefit to choose sample size for extraction and implantation experiments. The purpose of the test is to find out the response of animals towards the samples that are intended to fabricate a medical device or samples from the end product. So we have seen that the extraction mediums are sodium chloride injection, 1 in 20 solution of sodium chloride in ethyl alcohol in sodium chloride, polyethylene glycol 400, cottonseed oil or sesame oil IP grade. So 4 extraction mediums are there. The standard temperature of extraction is 70 degrees centigrade, and the toxicologist can choose the extraction temperature depending upon the need for time for extraction and the details related to physicochemical properties of the test sample. So the ISO, International Organization for Standard, specifies 4 different temperatures for extraction. That is something like 37 degrees, 50 degrees, 70 degrees and 121 degree centigrade. If the sample can withstand the temperature, or time is selected for 121 degree centigrade 1 hour. If the sample won't withstand the 70 degree centigrade for 24 hours, 50 degrees for 72 hours, and 37 degrees centigrade for 72 hours. Here also the toxicologist has got the freedom to choose depending upon the data available regarding the physicochemical properties of the test samples and his choice. And presently, good laboratory practices has to be positively implemented in a testing laboratory. And friends, this video describing in detail regarding the USP test will be a simple explanation because I presented testing of biomedical devices earlier, but in that the biocompatibility testing of major devices and biocompatibility test for USP tests are put together in that presentation. So here I think I have divided into 2. One as biocompatibility testing for medical devices and another one for testing of USP 6 classes. So friends, thank you for listening to me. Please do not forget to send your questions or call me. Be sure to attend the Time with Thebsian on biomedical devices every Friday. If you like the program, press like, share and subscribe to this video. Kindly let your friends know about this, share the video with them, and definitely discuss you people together and let me know your comments. Feel free to send your comments. Hope to meet you next Friday. Thank you very much for listening to me and watching this video.